Okay, here we go. Oh, there we go. So we're starting with the endocrine system. The endocrine system is composed of ductless glands that synthesize and secrete hormones. The hormones are then released into the blood and transported throughout the body. They bind to target cells that have specific receptors for that hormone, and then that target cell will respond. Hormones transport the target cells is done from being released into the interstitial fluid. So whatever gland or cell it is that's releasing the hormone, it will release it into the interstitial fluid. It will then enter the blood. It gets transported within the blood, either freely floating or bound to a carrier protein. It will randomly leave the blood and enter interstitial fluid again, and then it'll float through the interstitial fluid until it finds one of its target cells with a receptor to bind to. The endocrine and nervous systems are the two control systems of the body. We did nervous system in 168. Also, I'm 6'4", so I'm sorry. This is about as good of an angle as you're going to get on me. So the endocrine systems are the two control systems of the body. Both the endocrine and nervous system release ligands, which are those chemical messengers to help regulate or initiate some kind of response. Unlike the nervous system, the endocrine system transmits hormones through the blood. It targets any cells in the body with correct receptors, so that action can be very widespread. The effect can be very widespread, as opposed to the nervous system and neural cells, which have to interact with each other or at very specific locations. The endocrine system is going to exhibit longer reaction time, so as opposed to being very, very quick, even a prolonged nervous stimulus is still measured in seconds. The endocrine effects can have much longer times, both to be initiated and having this longer lasting effects, minutes to days to weeks. So these are the two communication methods that we see. So your nervous system releasing or having that electrochemical gradient, harnessing it to transmit an electrical signal at the synaptic knob, versus the endocrine system, which is going to release hormones directly to the blood to bind to target cells. Okay, so general functions of the endocrine system. Note on the notes, just because something is red does not mean that it's more important. It's just when I'm going through, I'm just highlighting certain words and blocking things out to make sure that you're engaging with the notes. It's not that these things are more important. Sometimes they are, sometimes they aren't, but don't put any specific weight to that. So general functions, you have the regulating development, growth, and metabolism. Hormones help regulate embryonic cell division and differentiation, as well as regulate metabolism, both anabolism and catabolism. All of our cell division, all the way through puberty, into adulthood, aging, and throughout our life is regulated by the hormones. Maintaining homeostasis of blood composition and volume. So hormones can regulate blood solute concentrations, such as glucose with insulin and glucagon, as well as ions with some of our mineralocorticoids. Hormones also regulate blood volume, cellular concentration, and platelet number. Blood volume can be regulated through how we sweat, our thirst centers, as well as through urine production. All of that can be controlled with hormones. Platelet numbers, as our blood gets thicker, it can be, we can release hormones to help thin it out, or if we need to increase our platelet count, we can use hormones to do that as well. Controlling digestive processes, hormones influence secretory processes and movement of materials in the digestive tract. Digestive regulation is pretty interesting. Your small intestine and your large intestine, your stomach all release different hormones to help regulate the movement of material. And that makes sense. You don't want to have your stomach emptying really quickly if your bowel is not emptying as quickly. So just to make sure that things don't get, there's no backing up happening, we can use hormones to help regulate that. As well as controlling reproductive activities. Hormones affect development of function of reproductive systems and the expression of sexual behaviors. Testosterone, estrogen, progesterone, FSH, LH, all of those hormones are going to be used for sexual development and reproduction. Sorry, let me make sure this isn't, my screen's falling over. Okay, so location of major endocrine glands. Your endocrine glands contain epithelial tissue that make and release hormones within a connective tissue framework. So endocrine glands contain epithelial tissue and connective tissue. Those are two of the tissue types that you did in 168. So your connective tissue framework, remember connective tissue provides support, protection for structures, and epithelial tissue is going to allow for secretion, absorption, those kind of things. So you have two different tissue types. That makes it a type of organ. You have to have more than one tissue type to have an organ. So we talk about endocrine glands and endocrine organs. So some glands are endocrine organs with sole endocrine functions. Those would be your pituitary, your pineal, thyroid, parathyroid, and adrenal glands. Those are solely endocrine organs. They are glands because they're secreting something. They're organs because they have two tissue types. So just a way to make you organize things even more. It's science. We like to organize it. You probably have heard of these, maybe not. You might have just heard of them, don't know their function as well. With this endocrine cells that are found in clusters in organs with another function, you've probably heard of a lot of these, such you've heard of all of these, and could give some example of some of their functions. Maybe not their endocrine function right away, but for the skin, the heart, the liver, the stomach, adipose connective tissue, kidneys, gonads, you know some of their functions. So we're going to get into their endocrine functions for this lecture. Here's just a visual representation of that. You have the major glands, the sole endocrine glands on this side, and the glands containing cells on the other side. Okay, so stimulation and hormone synthesis and release. So hormone release is regulated by reflexes to stimuli. Hormonal, humoral, or nervous stimuli can initiate a hormone release. So it's regulated by reflexes to stimuli. We saw that our nervous system also responds to stimuli. Do a little squat. And the endocrine system does as well. So there are three different types of stimulation that our endocrine system can have. Hormonal stimulation is when a glandular cell releases its hormone or when some other hormone binds to it. A humoral stimulation is a gland releases its hormone when there's a certain change in levels of a nutrient or ion in the blood. And then nervous stimulation is going to be when a neuron stimulates it. So the examples that you're commonly going to see are for humoral stimulation, something like TSH, thyroid stimulating hormone, is released from the anterior pituitary. It stimulates your thyroid gland to then release thyroid hormone into the blood to help regulate metabolic activities. So this is hormonal stimulation. You have one hormone, TSH, stimulating the release of another hormone. And actually TSH is stimulated to be released by TRH from the hypothalamus up here. So the hypothalamus releases TRH, that stimulates the anterior pituitary to release TSH, and then you have the thyroid gland releasing TH. And then this can go on and cause other hormones to be released as well. So that's your hormonal stimulation. One hormone stimulates the release of another. Humoral stimulation is saying that there's some type of change in blood chemistry. So remember, part of the endocrine system function is regulating homeostasis of blood chemistry. So here you might have some blood glucose levels either going up or down. So your body will release glucagon to increase your blood glucose levels, or insulin after a meal, after eating, to decrease your blood glucose levels. And then the third one is the nervous stimulation. So if the sympathetic division is activated, that will stimulate the adrenal glands to release epinephrine and norepinephrine to stimulate some neural cells. So that would be your nervous stimulation of the endocrine gland. They're releasing their hormones. That might then go on to hormonally stimulate something. Okay, circulating hormones. Now we're going to talk about circulating versus local hormones. There are three types of circulating hormones that are circulating through the blood. The first one is steroids. Steroids are lipid-soluble molecules. It's very important that you know that they are lipid-soluble. We'll get into the differences between lipid and water-soluble. They're pretty distinct. Lipid-soluble molecules synthesized from cholesterol. These include gonadal steroids like your estrogen, testosterone, as well as cortisol, synthesized by the adrenal cortex. Sometimes calcitriol is included in there. Calcitriol is for regulating blood calcium, but it's technically a sterol. Biogenic amines are modified amino acids. They include your catecholamines, your thyroid hormone, and your melatonin. Melatonin is the sleep hormone. You can take it to help you sleep as well. You can get supplemental melatonin, but some people say it gives you weird dreams, so be careful. Biogenic amines are water soluble except for your thyroid hormone. Your thyroid hormone is made from tyrosines, which are nonpolar. So your thyroid hormone is a lipid soluble biogenic amine. Know that. And last one is proteins. Most of the hormones are going to be proteins. These are water soluble chains of amino acids. Okay, local hormones. So those are the circulating hormones. Those are the ones that are going to be floating through the blood. The local hormones don't circulate in the blood. They bind to cells that release them, which is autocrine stimulation. So auto self. So the cell that releases it, it the hormone just gets released and then just binds right back to itself. And then or binds to neighboring cells, paracrine stimulation. Para meaning near. So it'll be released from one cell and then bind to one right next to it. Whoops. It is okay. Let's just set this back up. My dog got a little freaked out. It's all right. Hopefully I will get a steadier stand. Okay, so local hormones, icosanoids. So icosanoids are a type of local hormone formed from fatty acids within the phospholipid bilayer of the plasma membrane. So remember, the plasma membrane is made up of that phospholipid bilayer. It's synthesized, icosanoids are synthesized through an enzymatic cascade. Prostaglandins, you may have heard of prostaglandins, are a type of icosanoids. They have a variety of functions throughout the body. One of them is stimulating pain and inflammatory responses. If you take aspirin or any other kind of anti-inflammatory drugs, they block prostaglandin formation, so you do not feel the pain or inflammation. You don't experience the inflammation. This is important because for circulating hormones, let's say your hypothalamus and your brain is going to release a hormone that is going to be regulating your digestive tract. That is going to take some time. There's going to be some other influences that have to happen. For local hormones, this allows for local glands, local cells, local organs to self-regulate. To not have to wait for the whole for the whole endocrine system to communicate with itself to be able to release regulatory hormones. Icosanoids and local hormones are a way for a cell to be able to more quickly regulate itself and respond to stimuli. Okay, so that's why they're local. Here is that formation. You have an enzyme, phospholipase A2, so phospholipase, phospholipids, is going to remove arachidonic acid from the phospholipid membrane. So it takes the plasma membrane, it takes about a chunk of a 20-carbon fatty acid out of there, and then it goes through some other enzymatic chains, and you get prostaglandins, thromboxans, and leukotrienes. 3 different types of icosanoids. Okay, so now we're doing transport in the blood. So those were the local ones. These are back to the circulating. Remember we said we were talking about the difference between lipid and water soluble. So lipid soluble hormones use carrier molecules to be transported through the blood because they do not readily dissolve in blood. Carriers are water soluble proteins made by the liver, and they protect the hormones from early destruction. So they have they allow what would be a lipid-soluble molecule, or what is a lipid-soluble molecule that is not going to dissolve in the highly water-concentrated blood. So they have these proteins that are basically just going to kind of bind to them that allow them to just be transported through the hydrophilic blood. The binding between the hormone and the carrier is temporary. It is not a long-term thing. There is attachment, detachment, and reattachment are common. So it will bind, it will release, it will bind, it will release to carrier proteins as it's going in and out of the bloodstream and interstitial fluid. Most of the hormone, 90% or more, is bound hormone that's transporting through the blood. Only unbound, free hormone is able to exit blood and bind to target cell receptors. Most water-soluble hormones travel freely through the blood, and a few use carrier proteins to prolong their life because it does allow them to have a longer lifespan. But back to this point about only unbound hormone is able to exit the blood and bind to target cell receptors. This is speaking about lipid-soluble hormones. So for a lipid-soluble hormone to actually produce its effect, it has to unbind from the carrier protein and then exit into the interstitial fluid. I'm sorry, unbind from the protein and then bind to the target cell. Okay, so levels of circulating hormone. A hormone's blood concentration depends on how fast it is synthesized and eliminated. So synthesized, how fast it's made, and how fast it is removed. We'll see that eliminated does not just mean you excrete it through your urine or feces. We'll see that eliminated can also mean how fast it's uptook. So hormone release and its concentration in the blood are positively correlated. An increase in release results in higher blood concentration and vice versa. So it's just saying that as you release more hormone, its concentration in your blood are going to increase. Makes sense. Hormone elimination occurs in multiple ways. You can have enzymatic degradation in your liver cells. You can have removal from blood via the kidney excretion or target cell uptake, meaning that the cell that's supposed to bind to it will bind it and remove that hormone from the bloodstream, which would decrease your blood levels. The faster the elimination rate, the lower the blood concentration. So the faster it can be taken up, the faster that your kidneys can excrete it, your liver can break it down, the lower your blood concentration will be. Sometimes as your blood concentration lowers, that signals that you need to have more synthesized and more made. The half-life is the time necessary to reduce a hormone's concentration to half of its original level. So half the original level. If you're start off releasing 100 milligrams of a hormone, by the time it gets to 50, however long that is, that's its half-life. Half-life depends on how efficiently it's eliminated. Hormones with short half-life must be secreted frequently to maintain normal concentration. So if you're having something that's secreted frequently, then it's going to have to constantly be made again. Water-soluble hormones generally have shorter half-lives. Again, because they're being dissolved in the body, they don't have that carrier protein protection. They tend to have a shorter half-life, a few minutes for some small peptide hormones. Steroids generally have longer half-lives because they have the carrier protein providing protection. Your testosterone half-life is 12 days, pretty long. So lipid-soluble hormones, let's get into their specific effects. So lipid-soluble hormones can diffuse across target cell membranes. So when they are being transported through let me not get ahead of myself. I always get ahead of myself and I need to just read the slides. So such hormones are small, nonpolar, and lipophilic, so meaning that they are fat-loving. The receptors are in the cytosol or nucleus. In the cytosol or nucleus, not on the membrane, but they're inside of the cell. Once the hormone enters the cell, it binds to the receptor and forms a hormone receptor complex, an HRC, hormone receptor complex. This complex then binds to a hormone response element, your HRE, on your DNA. The HRE is a segment of DNA that is going to be bound by this hormone response element. Let me see if my pointer works. It kind of does. You see that little black fly-looking thing? That's my pointer. I'm not good at that. So your hormone response element is going to bind to that segment of DNA. This is going to initiate transcription of a certain protein. So it's going to result in transcription of an mRNA, which is translated to a protein, and the protein may have structural or metabolic effects. So what you have is a lipid-soluble hormone bound to a carrier protein floating through the blood. It's going to randomly exit. It's going to be unbound. It's going to exit out, bind to its target cell, and then it's going to diffuse across that target cell membrane until it can then form this hormone receptor complex. That complex will again find a segment of DNA that codes for a certain protein, and it will initiate that protein translation. Here is just a, if you like, kind of a picture illustrated view of it. So yeah, again, you have the unbound protein or unbound hormone diffusing into the cell. That then goes into the nucleus. It binds to that segment on the DNA, which then causes transcription of a protein. Anytime, or generally, when we talk about a segment of DNA, we're going to call it an element. It's just one of those weird biology things. I know elements are, you think chemistry, but for in biology, a lot of times when we're talking about a segment of DNA, we're talking about an element. I don't know. Well, I didn't name it that. So water-soluble hormones. So water-soluble hormones are going to use membrane receptors on the plasma membrane. Remember, it's a phospholipid membrane. Water-soluble is not going to be able to diffuse very easily. A water-soluble hormone is not going to be able to easily diffuse through a big phospholipid membrane. So it's going to stay outside. These hormones are polar and they can't diffuse through the membrane. They're going to induce what's called a signal transduction pathway. Okay, so a signal transduction pathway is used to activate some larger response within a cell. With lipid-soluble hormones, we were trying to synthesize proteins to try and, I'm trying not to get like, there you go, now you can see me. We were trying to synthesize lipid-soluble proteins so that way we could, or we were using lipid-soluble hormones to synthesize proteins for metabolic effects or regulatory effects. In water-soluble hormones, we're going to be creating what we call signal transduction pathways to have this cascade of events to have bigger, more widespread effects. So in a signal transduction pathway, your hormone is your first messenger. It initiates the events by binding to a receptor. So it binds to the membrane receptor on the plasma membrane of your target cell. When it binds, it activates what's called a G protein. It was mentioned in 168. You may not remember it, but it was mentioned, especially if you took my class. Binding activates a G protein, which is an internal membrane protein that binds a guanine nucleotide. Remember, there are 4 nucleotides. So it binds a guanine nucleotide. Activation results in the binding of GTP instead of GDP. So it basically is just an activation of GTP and ATP are energy molecules. It's the same thing. ATP is more commonly used and it's higher energy than GTP. But in this cascade, you have your hormone binding. Your hormone is your first messenger. We'll see that there are first, second, and sometimes third messengers. So your hormone is always your first messenger in this signal transduction pathway. It's going to then activate this G protein, which is then going to activate either the adenylate cyclase or phospholipase C pathway. Okay, so it can activate one or the other. The activated enzyme catalyzes the formation of a second messenger, a chemical that modifies cellular activity. So here is the activation of your G protein. You can see that up here you have your water-soluble molecule binding to the receptor protein with your inactive G protein in here. Once the water-soluble molecule binds to the receptor protein, it's going to go through the receptor protein is going to go through a conformational change. That conformational change is then going to make available this binding site right here. And this binding site is going to allow GDP to turn into GTP. Once that happens, the G protein is then activated. Once it now has GTP bound to it, it's going to be activated. That's then going to slide down. And the first one that it might activate is the adenylate cyclase. So after the hormone, depending on the hormone, if it's glucagon, binds to its receptor, the G protein is activated. That protein will slide down the plasma membrane and activate the adenylate cyclase. So cyclase, it's an enzyme. It's an enzymatic pathway. Adenylate cyclase generates cyclic AMP. So we've seen that before. It was mentioned definitely when we did, or I say we because we are all in this together, when you studied the senses. So talking about light production. Actually, I think it was cyclic GMP, but it's the same things. So you have adenylate cyclase generating cyclic AMP. Cyclic AMP activates a protein kinase. Remember, kinases are going to add phosphate on and off of things to help activate or deactivate it. Protein kinase A phosphorylates other molecules, activating or inhibiting them. There you go. So this is that adenylate cyclase activity. It's going to start with an activated G protein that activates your adenylate cyclase enzyme. That'll start to generate cyclic AMP. Cyclic AMP activates protein kinase. That kinase is going to then go forth and activate or inhibit a large array of things. So it's going to have this amplified effect. Here is just that rendered depiction of that if you want to see it in your book. Okay, so phospholipase C. So let's say that the hormone is epinephrine. It's going to bind to a receptor. Your G protein gets activated. That G protein is then going to activate the phospholipase C. Here's where it gets interesting. Here's where it gets different. Your phospholipase C, again, lipase, it's an enzyme. Your phospholipase C splits this PIP2, which is a name you don't have to know. All you need to know is PIP2. You don't have to know the full name. It's going to split PIP2 into DAG and IP3. These DAG and IP3 are going to be the second messengers. Okay. The DAG will activate your protein kinase C. That is going to be a phosphorylating enzyme. Your IP3 is a second messenger that leaves the membrane and causes an increase in the levels of calcium in the cytosol. That increase is caused by effects on the endoplasmic reticulum within the cell and calcium channels, membrane calcium channels. Your calcium is then going to be a third messenger activating kinases, sometimes calmodulin for smooth muscle contractions, and interacting with ion channels. Okay, so that one's a little bit more tricky because we have the second and the third messengers. So here's this image of it. So you have your G protein activating your phospholipase C. Remember, your phospholipase C is going to take part of this phospholipid chain. It's going to split your PIP2 into DAG and IP3. These are two different pathways at this point. So they're both second messengers. DAG and IP3 are second messengers. DAG is going to activate protein kinase C, which is then going to go on and phosphorylate different things and cause different reactions. So the DAG pathway stopped at a second messenger. The IP3 pathway is then going to act on your endoplasmic reticulum, your calcium channels, your calcium pumps, and it's going to then initiate calcium as a third messenger. Okay, so you still start with the G protein activating phospholipase C, splits PIP2 into DAG and IP3. Those are your second messengers. Calcium is your third messenger, only initiated by IP3. If it doesn't have IP3, you're not going to have calcium release. DAG can't also release calcium. IP3 can't also activate the kinase. They have to go through their pathways. Okay, water-soluble hormones. With water-soluble hormones, you have these multiple results possible with different signal transduction pathways. Okay, those two different pathways. Your enzymes can be activated or inhibited. You can have growth can be stimulated for cell division. Cellular secretions can be released. Membrane permeability can be changed. Muscles can be contracted or relaxed. So you have this wide range of potential effects from water-soluble hormones. The intracellular enzyme cascade, your signal is amplified at each enzymatic step. Just a few hormones, hormone molecules can change many molecules within the cell, and there are many places to regulate pathway activities through this cascade. You have a lot of the different G proteins can be activated. You can have multiple G proteins activated. Just going back real quick, when we were looking at it, we say it's like, okay, you have one G protein, you have one phospholipase C. It's not really how it works. That water-soluble hormone, I'm going to go all the way back here. Here we go. This water-soluble hormone will stay bound until there's some other signal that kicks it out, until there's some other hormone, probably that binds to this activated receptor to deactivate it and kick the water-soluble hormone out. So as long as this hormone is bound to the receptor, it's going to continuously activate G proteins, which are going to go through and continuously activate those cascades. You're generating cyclic AMP, you're generating calcium, you have all these. I'm pointing my laser pointer at my computer, like y'all can see that. You have these PIP2, you have the DAG and the IP3 being formed over and over again, activating different their different pathways. So the signal is amplified at many steps, many places to regulate it. The cells possess mechanisms to quickly inactivate intermediates and break down second messengers. So once the stimulus has been responded to, the body can then start to break down those second messengers. Okay, degree of cellular response. A cell's response to a hormone varies with its number of receptors and its simultaneous release to other response to other hormones. So the more receptors it has, the more sensitive it will be to that hormone. And its response to other hormones. It's not like there is just one and then another hormone is going to wait its turn and that will bind. You have hormones constantly competing, responding, and adjusting to stimuli to maintain homeostasis. Receptor number fluctuates. Upregulation increases the number of receptors and increases the sensitivity to a hormone. Sometimes that occurs when blood levels of hormones are low, or sometimes it occurs with changes in development, cell cycle, and cell activity. So let's say during puberty, during times of growth, especially as we're younger, as we age, we have less cycles of growth. But our cells may upregulate their number of receptors for thyroid hormone, which would then allow them to bind more thyroid hormone, which will initiate even more metabolic responses. If a cell has more receptors for insulin, it will be able to bind more insulin and be able to break down glucose even more. That's how it works. Downregulation is going to then decrease the number of receptors. It will decrease sensitivity to a hormone. Sometimes it occurs when blood levels of hormone are too high. So let's say you're releasing too much insulin. Your body you are releasing excessive amounts of insulin. Your cells might start to downregulate their number of receptors because they can have a fewer number of receptors and still have a sufficient response to stimuli because there's so much of the hormone being released. It also occurs with changes, same thing, development, cell cycle, and cell activity. As we go through life, we go through periods of growth and development. As we get out of those, we start to downregulate receptors to conserve energy. And here's just a picture of that. Okay, hormone interactions on a target cell. So different hormones can simultaneously bind to a cell and their effects may interact. So two hormones or more binding to one cell. So insulin and glucagon. Let me read the examples. So synergistic interactions. One hormone reinforces the activity of another one. So this is going to be synergistic. They're going to be working together here. Examples: estrogen and progesterone effects on a target cell. Right. So they're both going to be having the same effect. Permissive interactions. You have one hormone requires the activity of another hormone. For oxytocin's milk ejection effect requires prolactin's milk generating effect. So prolactin is what is the hormone that allows for synthesis of milk, and then oxytocin allows for release of milk, what we call the letdown of milk. So this is almost a hormonal one, a hormonal stimulation. Antagonistic interactions. One hormone opposes the activity of another hormone. So glucagon increases blood glucose while insulin lowers it. So three different ways that you can have hormones regulating target cells. This is just that in a graphic form for people who like graphic forms. So these next slides, they look a little overwhelming, I know, but don't freak out. These are just overviews of pretty much everything that we've done so far. We've already covered all of this. So you should be able to look at these pages. If you break it down into block by block, this is identifying the different cells and glands, the different types of transport, the different effects of water versus lipid soluble, and regulating interactions and number of receptors. So this is a great little page to study with, just to give you, again, an overview of what we did. And this is just breaking it down. Cool. So let's talk about the hypothalamus and the pituitary gland. So getting into some specific glands and organs here. So the hypothalamus controls the pituitary gland, which controls your thyroid, adrenal, liver, testes, and ovaries. Your pituitary gland is a hypophysis. It lies inferior to the hypothalamus in the cella turcica of the sphenoid bone. So it lies inferior to your hypothalamus, and the cella turcica, which is that little part of your frontal lobe that has looks like a butterfly almost, has wings out to the side. It's about pea-sized. It's connected to your hypothalamus by an infundibulum and partitioned into anterior and posterior pituitary. We'll see they have structural and functional differences. Your posterior pituitary is a neurohypophysis. So we'll see that it's the neural part of the pituitary gland. It's also smaller. What you have are hypothalamic neurons project through the infundibulum, through the stalk connecting the hypothalamus and the pituitary gland, and release hormones in the posterior pituitary. We'll get into this, but essentially the hormones are formed in the hypothalamus. They're formed in the hypothalamus, and then they're transported and stored in the posterior pituitary gland. Your posterior pituitary is going to be storing ADH and oxytocin, antidiuretic hormone, which helps to decrease urine production, and oxytocin, which we'll see plays a role for, we already saw, does milk production, also in uterine contractions. So this is your posterior pituitary. You have these neural projections. Again, this is the neural part of the hypothalamus. Here is your anterior pituitary, and then this is your infundibulum that is connecting them to your hypothalamus. There we go. Which is just a region. Your hypothalamus is a general region right here in your brain. The anterior pituitary. The anterior pituitary has the hypothalamo-hypophyseal portal system of blood vessels connecting the hypothalamus to the anterior pituitary. When you see portal system, similar to how you saw element, you should think of just a region of DNA. When you see portal system, you should think of some type of vessels, some type of transport tubes. So hypothalamo-hypophyseal portal system connects the hypothalamus to the anterior pituitary. It has three portions: the primary, the secondary plexus, and the hypophyseal portal veins. The primary plexus, a plexus is just a network of nerves or vessels. Doesn't matter what it is, it's just a network of them. So the primary plexus is a porous capillary network associated with the hypothalamus. So this is going to be surrounding the hypothalamus. The secondary plexus is associated with the anterior pituitary. And then the hypophyseal portal veins drain the primary plexus and transport to the secondary plexus. So here you can see these plexi, the nerves just again wrapped around these portions of the infundibulum and the anterior pituitary. What happens here is that the hypothalamus is going to synthesize and release, we'll see a lot of what we call regulatory hormones, and then those are going to then stimulate the anterior pituitary gland to release or stop releasing its hormones. So the hypothalamus has TRH, PRH, PIH, GnRH, CRH, and GRH. You should know all those. Let's go ahead and put that out there. Any of the hormones, you should know them. Your thyrotropin releasing hormone, inhibiting hormone, go ahead and kind of connect those things. If it's releasing hormone, it's going to stimulate hormones to be released. And if it's an inhibiting hormone, it's going to inhibit that hormone's release. And these are very straightforward. So your prolactin inhibiting hormone is released from the hypothalamus, and it inhibits the release of prolactin from the pituitary gland. Again, I think asking you to know these is not a huge stretch for some of them. Your anterior pituitary, it synthesizes and releases thyroid stimulating hormone. We've seen that one already. Prolactin. You have follicle stimulating hormone, FSH and LH. You have ACTH and GH, your growth hormone. So these are all synthesized and released in the anterior pituitary. Remember, the posterior pituitary does not synthesize its hormones. It stores hormones from the hypothalamus and releases them when signaled. Okay, interactions between the hypothalamus and the posterior pituitary gland. So your posterior pituitary is a storage and release site for oxytocin and antidiuretic hormones. They're made in the hypothalamus by neurosecretory cells packed in secretory vesicles and transported by fast axonal transport, if you remember your types of neural transport. They're then released from the synaptic knobs into the blood when neurons fire impulses. Your ADH, it's also known as vasopressin, so antidiuretic hormone, ADH, vasopressin. If I call it or refer to it as vasopressin, I will also add the ADH and antidiuretic hormone to it as well. It's made in the supraoptic nucleus. Its functions to decrease urine production, stimulate thirst, and constrict blood vessels. Overall, this will help to raise your blood volume levels and raise your blood pressure. If you're decreasing urine production and stimulating thirst, you're going to make yourself drink more and hold on to more urine. That will increase your blood volume. And constricting blood vessels constricting vessels increases restriction. That will increase blood pressure. Also, increasing blood volume increases blood pressure. Oxytocin, it's made in the paraventricular nucleus. It functions for uterine contraction, milk ejection, and emotional bonding. Your hypothalamus hormonally stimulates the anterior pituitary to release its hormones. The hypothalamus secretes its regulatory hormones that travel through those portal vessels to the pituitary gland. The anterior pituitary secretes hormones into general circulation. Okay, so the hypothalamus does not secrete hormones into general circulation. It's going to be leaving that up to the pituitary gland. So the hypothalamus, hormones of the hypothalamus, you're releasing hormones and you're inhibiting hormones. Releasing hormones increase secretion of the anterior pituitary, whereas you're inhibiting decrease the secretion. You should know all of these. Thyrotropin releasing hormone. Tropic refers to growth. So thyrotropin releasing hormone is talking about releasing. It's a hormone that's going to cause the release of a hormone causing the growth of the thyroid, if that makes sense. So thyrotropic refers to growth of the thyroid. TRH stimulates the release of TSH, which is thyroid stimulating hormone. So you can see how that cascade proceeds. Okay, in the anterior pituitary, you have TSH, which is released triggered from TRH, causes the release of TH from the thyroid gland. PRL is your prolactin, it's triggered by PRH or inhibited by PIH, causes milk production, mammary gland growth in females. Adrenocorticotropic hormone, your ACTH, it's a corticotropin. It's triggered by CRH, corticotropin releasing hormone, and it causes release of corticosteroids by the adrenal cortex. Gonadotropins, your FSH and your LH. So gonadotropins, these are going to help with the growth of your gonads essentially. So your follicle stimulating hormone, your luteinizing hormone help with sperm and egg production. In females, regulate ovarian development and secretion of estrogen and progesterone. In males, sperm development and secretion of testosterone. The anterior pituitary also releases growth hormone. It's called GH and somatotropin. Again, if you think somatotropin, so growth, somatic body, so growth of the body, so growth hormone. It causes the liver to secrete insulin-like growth factors 1 and 2. So your growth hormone and IGFs, your insulin growth factors, function synergistically, so synergistically function together to stimulate cell growth and division. There is a graphic representation. Okay, so growth hormone, its effects and its regulation and effects. So regulation of growth hormone release is controlled through hormonal stimulation, GHRH and GHIH from the hypothalamus, so release hormone or inhibiting hormone. The amount of GHRH released from the hypothalamus is impacted by your age, the time of day, the nutrient levels, and your stress and exercise. So all 4 of these are going to play an important role in regulating how much or how little growth hormone is going to be released. I think we should get more in depth into these, just in case we don't. So as you age, you release less. Throughout the time of day, that will just kind of go on a cycle. That's all about your circadian rhythms and making sure that your body is on a natural and normal, healthy wake-sleep cycles. Nutrient levels, as you have more nutrients, your body will be signaled to deposit more and create more growth. As you have less nutrients, your body will start breaking things down and stop growth and repair. That's why it's important to have, calories aren't just calories. You need to have a well-balanced diet, so that way you actually have the nutrient levels necessary to fuel growth and repair. If you don't, then your body will start releasing more GHIH to limit the type of repair and growth that's happening. In stress and exercise, moderate, low levels of stress and exercise are good. They help your body to release more GHRH to repair itself. Excess of either will cause for proteins to be broken down and your body to start breaking itself down. I don't have to tell you that's bad. You don't have to finish this course to know that your body breaking itself down is bad. Okay, so effects of growth hormone. So we already talked about how it stimulates the insulin-like growth factors, the IGFs from the liver. These are similar to your growth hormone, but they have a longer half-life, so they will allow to prolong its effects. All cells have receptors for GH, IGFs, or both. So all cells, think about that, all of your cells, except for a couple, of course we have to qualify that, but all cells have receptors for these types of hormones. Hormones stimulate increased protein synthesis, cell division, and cell differentiation, so growth and repair. It also stimulates release of nutrients from storage. You should be able to know and identify all of these terms: glycogenolysis, gluconeogenesis, glycogenesis, lipolysis, and lipogenesis. You have glycogenolysis, breakdown of glycogen into glucose. This is going to increase your blood glucose. You're going from its stored form into its active form. Gluconeogenesis. Neogenesis is the creation of glucogen. Is going. Gluconeogenesis. Sorry, is the creation of glucose from some type of new source. So it's going to convert nutrients, fats, proteins into glucose. Glycogenesis, that's what I was thinking of. Glycogenesis is the synthesis of glycogen, lipolysis, breakdown of lipids, your triglycerides, and lipogenesis is the formation of triglycerides for storing energy. Here is a nice little chart for that. So this is showing, again, those variance in levels. You can see that during the times of day, it all just kind of goes up and down. All right, clinical views. So these are good. I don't, my background is not in clinical medicine, so I don't get too in-depth into clinical stuff, but I do like to go ahead and introduce it. I'm gonna go ahead and start showing you some of it. So your growth hormone deficiency, pituitary dwarfism. If you have a deficient amount of growth hormone, you have, due to hypothalamic or pituitary problems, you end up with short stature and lower blood sugars. And these are going to be, varied. There's a scale, there's a wide spectrum that you could be affected with these. Pituitary gigantism is too much growth hormone, excessive growth and increased blood sugar, enormous internal organs, and death at early age if untreated. So I am just tall. I do not have pituitary gigantism. Acromegaly, I also don't have this, is excessive growth hormone production as an adult. So your pituitary gigantism is going to be as a child. This is going to be as an adult. Enlargements of bones, face, hands, and feet, increased release of glucose, internal organs increase in size, and results from loss of feedback control of growth hormone. So your body is no longer able to inhibit those effects of growth hormone and IGFs. All right, so now getting into the thyroid gland. So the thyroid gland is inferior to the thyroid cartilage of the larynx, anterior to the trachea, so just right on the front of the throat. It's divided into left and right lobes. Unlike the pituitary gland, the left and right don't have any type of functional difference. They're or structural rather. They're connected at the midline by a narrow isthmus. So you have the isthmus that connects the two lobes. There's rich vascularization, which gives it its reddish color, and it's composed of microscopic follicles. There are follicular cells and parafollicular cells. The follicular cells are cuboidal epithelial cells that surround a central lumen and synthesize thyroglobulin. Okay? This is going to produce and release thyroid hormone. The follicle lumen houses colloid, which is a viscous protein-rich fluid. We'll see this word colloid a lot through these first few chapters, especially as we talk about the blood and things. Colloid is just a protein-rich fluid. Don't let it get you any more confused than that. When you see the word colloid, it just means that it's a fluid with a bunch of proteins in it. So the follicle lumen houses this colloid, this protein-rich fluid, and then your parafollicular cells are the cells between the follicles making calcitonin, which decreases blood calcium levels. So this has within your thyroid, you're going to have follicular cells producing and releasing thyroid hormone. You're going to have colloid in the lumen of the follicular cells. And then you're going to have parafollicular cells. They're going to be producing calcitonin. There's a picture of it. You can see it is nice, bright and red. It's this thing right there. There's your larynx. Oh, yes, here we go. So these are... right in here is going to be the colloid fluid. This is that inside of the follicular cell. You can see kind of the cells surrounding it. They're going to be your parafollicular ones, a couple of them. So here is the synthesis, storage, and release of thyroid hormone. I think, no, it doesn't. Okay, so you should know these steps, and you should know the phases of thyroid hormone production. First, you have an iodine ion uptake. So right over here, you can see this active transport of iodine ion that's just circulating the blood. That's why we have iodized salt. If you don't, you get a goiter on your throat because you need to have this iodide to be able to synthesize and release thyroid hormone. So that gets transported in. As the iodide gets transported in, it turns into iodine. So you have two iodide molecules joined together to form iodine. And then you have your thyroglobulin synthesis. So while you're having this formed, your thyroglobulin molecules are synthesized within the rough ER, and they're shipped to the Golgi apparatus for modification, and then transported by vesicles and released from the follicular cell into the colloid fluid. So this is your bloodstream. This is the inside of the follicular cell. So this is the inside that we're looking at right in here. And once you get inside your follicular cell, the thyroglobulin molecules with the iodine molecules attached to its tyrosine amino acids are endocytosed into a follicular cell. So you have these iodine and these thyroglobulin molecules joining together, and then they are transported back into the follicular cell. Then it's going to make T3 and T4. So a vesicle-containing thyroglobulin with attached pre-T3 and pre-T4 fuses with a lysosome. Lysosomal enzymes cleave pre-T3 and T4 from the thyroglobulin to release just T3 and T4, and the thyroglobulin is resynthesized. So those last, that's 5 and 6, that's probably a little confusing. That's why we're going to jump into this. So real quick though, you have iodine being taken in, it gets transported into the colloid with thyroglobulin. There, those two join and then are brought back into the cell. Once they're brought into the cell, they have pre-T3 and pre-T4 bound to the thyroglobulin. The thyroglobulin gets cleaved off and then recycled, and the T3 and T4 can be released into the bloodstream as thyroid hormone. Thyroid hormone comes as both T3 and T4 varieties. So here's kind of just generally how that happens. You have this thyroglobulin protein right here. You have a tyrosine, in this position, and you have these 4 iodine molecules. There's an enzymatic reaction that happens that binds the iodines to the tyrosines. And then another enzymatic reaction, you have a peroxidase that is going to then kind of close this off and make T4. This is T4 because there's 4 iodines. If only 3 get bound, then it's T3. That's really the only difference. It's just a matter of our 4 or 3 iodines binding. Okay, regulation of thyroid hormone release. So hypothalamic pituitary thyroid axis. Your hypothalamic, hypothalamus, pituitary, anterior, posterior pituitary, thyroid. So remember, these are all regulating with each other, or rather, these regulate down. Cold temperature, pregnancy, high altitude hypoglycemia, that's low blood sugar, hypoglycemia, or low thyroid hormone cause hypothalamus to release TRH. TRH causes the release of TSH. TSH binds to receptors of the follicular cells, triggering the release of TH. Follicular cells release 2 forms of TH into the blood, the T3 and T4. The T3, like I said, is the triiodothyronine, and T4 is tetraiodothyronine. They're the same thing. Let me rephrase. They're not exactly the same thing. T3 is more active. So once cells take up T4, they convert it to T3. Here is a pathway of that. So you can also see some of the effects that it has on organs. Okay, it's cellular transport brings thyroid hormone into the target cells, and it binds receptors. Your T3 versus T4, again, T3 is the more active form. T4 is more common though, so it usually gets converted. TH increases the metabolic rate and protein synthesis in target cells. It stimulates the synthesis of sodium-potassium pumps in neurons, so it's going to help to maintain the sodium-potassium gradient. It has a calorigenic effect to generate heat and raise temperature. It stimulates increased amino acid and glucose uptake, as well as increased numbers of cellular respiration enzymes within mitochondria. If you take all of these things together, you can see that it's going to increase protein production, increase growth. If you're having increased sodium-potassium pumps, you're going to increase your neurological activity. If you're stimulating amino acid and glucose uptake, that's both energy and building blocks for proteins. And then numbers of cellular respiration. Anytime you see cellular respiration or oxygen being increased, you should immediately connect that to increased metabolic activity. Okay, it fosters energy production, ATP production. So hepatocytes, your liver cells, stimulated to release to increase blood glucose. So your thyroid hormone causes the increase in glycogenolysis and gluconeogenesis, and a decrease in glycogenesis. So glycogenesis being the production of glycogen from glucose, which is storing glucose and decreasing the levels. If you increase your glycogen, you're increasing. If you increase your glycogen storage, you're decreasing your glucose circulation, and that decreases the amount of energy that you have available. Your adipose cells are stimulated to increase blood glycerol and fatty acids, so you're not going to be forming as much fat. You're going to be actually inducing lipogenesis, and this saves glucose for the brain, the glucose-sparing effect. So your blood has to run on glucose. It needs that fast, quick energy. Thyroid hormone increases your respiration rate to meet additional oxygen demand, and the thyroid hormone increases the heart rate and force of contraction to help increase blood flow, to deliver more nutrients, more oxygen, more glucose. And it causes the heart to increase receptors for epinephrine and norepinephrine so that it can even further increase and continue that increase of heart rate. Okay, hypo versus hyperthyroidism. Hyperthyroidism, it's excessive production of TH. You have increased metabolic rate, weight loss, hyperactivity, and heat intolerance. Caused by T4 ingestion, excessive stimulation by pituitary gland, or loss of feedback control. That's the Graves' disease, is loss of feedback control. You treat it by removing the thyroid and giving hormone supplements. It can be tricky to try and figure out what the proper dose for thyroid supplement is for a specific person. It can take months and years to figure it out. Hypothyroidism results from decreased production of TH. You have a lower metabolic rate, lethargy, cold intolerance, and weight gain. Decreased iodine uptake, loss of pituitary stimulation, post-surgical immune system destruction of the thyroid, so that's Hashimoto thyroiditis. and treated with thyroid hormone replacement. Also knew someone who had a tumor in his thyroid gland, and so once they removed that, he immediately lost all of his symptoms. Goiter is an enlargement of the thyroid, typically due to insufficient dietary iodine. Lack of dietary iodine prevents thyroid from producing thyroid hormone. Once relatively common in the United States, but no longer now that iodine is added to table salt. So you get it. It's kind of like fluoride. You're going to get it, plenty of it. You don't have to look out for it. Calcitonin. Calcitonin is synthesized and released from the parafollicular cells of the thyroid gland. Remember, there was the follicular cells releasing thyroid hormone, parafollicular cells releasing the calcitonin. Its stimulus for release is high blood calcium or stress from exercise. It acts to decrease the blood calcium levels by inhibiting osteoclast activity or stimulating kidneys to increase excretion of calcium in urine. So if you remember, your osteoclasts are going to break down your bone cells. If you inhibit their activity, if you're inhibiting the breakdown of your bone cells, you're not only decreasing the levels of calcium in your blood, you're keeping and increasing the levels of calcium in your bones. That's why it's very important for people, especially women as they age, to keep up some moderate amount of exercise. Elderly people are very, very much encouraged to get up, to walk around, to move for 30 minutes, 20 to 30 minutes a day if they can, a couple of times a week or every day if possible, because that soft walking is good stress on the body, and it's that kind of stress. When we think stress, we immediately think something. Take your pick. But this kind of stress is talking about the actual physical stress of the impact on the body, on the bones, is going to cause an increase in calcitonin to decrease osteoclast activity. As similarly, stimulating kidneys to increase excretion of calcium in urine, that just allows for more calcium to be released. Wait a second. Yeah, because you have high blood calcium. All right, the adrenal glands. The adrenal glands are also called the suprarenal glands. Renal referring to your kidneys, supra meaning above. So your adrenal glands are suprarenal glands located on the superior surface of each kidney. Retroperitoneal, that refers to the position in the abdomen. Embedded in fat and fascia. So these are very much protected, is what you should understand from this. There are two regions. You have the adrenal medulla and the adrenal cortex. Medulla is in the middle. Medulla, middle. So this forms the inner core of each adrenal gland. It has a reddish-brown color due to extensive blood vessels, and it releases epinephrine and norepinephrine with sympathetic stimulation. Okay. Your cortex is going to synthesize more than 25 different corticosteroids. It has a yellowish color because it has lipids. And there are three regions to your adrenal cortex. You have your zona glomerulosa, fasciculata, and the inner zona reticularis. So there are your kidneys. You can see your kidney here. And then sitting right on top of it, this little flap, is the adrenal gland. You have one on each kidney. Here are the layers. So this is the inner medulla. And then this out here is the cortex, and it has the capsule surrounding all of it. You can see, let's see if we can break it down into, again, the capsule. You have this smaller area is the glomerulosa, this larger middle section is the fasciculata, and then this deeper section is the reticularis, and then you go into the medulla. All right, from the cortex, from the outer portion, we get mineralocorticoids, glucocorticoids, and gonadocorticoids. Mineralocorticoids regulate electrolyte levels. So minerals, electrolytes, hopefully you can connect those two. These are made in the zona glomerulosa, that thin outer layer. Aldosterone specifically fosters sodium retention and potassium secretion. So you should know the hormone, where it's released from, and what it does. So aldosterone fosters sodium retention and potassium secretion. Your glucocorticoids regulate your blood sugar. So glucocorticoids, hormones that regulate blood sugar. These are made in zona fasciculata, the larger middle area, and they include cortisol, which helps to increase your blood sugar. Okay? I know a lot of times when we think regulating blood sugar, insulin and glucagon do it more directly. Cortisol is going to have a more indirect effect. It's not going to so much bind to act like insulin does. Your gonadocorticoids are your sex hormones. They're made in the inner zona reticularis. They're called the androgens, or male sex hormones made by the adrenals, converted to estrogen in females. And the amount produced by adrenals is less than amount from the testes. So not as big of a production of androgens. Also, real quick, never mind. All right, regulation of cortisol release. Cortisol and corticosteroid increase nutrient levels in the blood to resist stress and repair injured tissue. So in times of moderate stress and acute stress and repair, cortisol and corticosteroid are going to increase blood nutrient levels to help for repair and growth. The release is regulated by the hypothalamic pituitary adrenal axis. Stress, late stages of sleep, and low levels of cortisol stimulate hypothalamus to release CRH, the cortisol releasing hormone, corticosteroid releasing hormone, sorry. CRH stimulates the anterior pituitary to release ACTH. ACTH stimulates the adrenal cortex to release cortisol and corticosteroid. So you have this hormonal stimulation pathway, this permissive pathway too. Cortisol travels through blood attached to other carrier proteins. Small amounts of cortisol dissociate from the carrier and leave the bloodstream. And then the cortisol levels are regulated by negative feedback. Cortisol inhibits the release of CRH from the hypothalamus and ACTH from the anterior pituitary. So as you have increased levels of cortisol, it's actually going to start to inhibit up its pathway at the hypothalamus and the anterior pituitary. Because if you had too much, then you're going to, too much cortisol and too much corticosteroid, then it's going to start to break down and keep nutrient levels too high. There's a graph, variable levels. You can see how sleep affects it in time of day. Increased stress increases cortisol. So cortisol causes target cells to increase blood nutrient levels. Liver cells increase their production of glucose, decrease glycogen storage. Adipose cells increase lipolysis. Many body cells break down proteins and back into amino acids. The amino acids can then be used to make glucose in the liver. And most cells are going to decrease their glucose uptake, sparing it for the brain. You can offer therapeutic doses of corticosteroid that's used to treat for inflammation. It inhibits inflammatory agents and suppresses immune system. At high doses, it does have side effects. It increases risk of infections and cancer, retention of sodium in water, and inhibits connective tissue repair. This is why if you have ever had some type of joint injury and you go and you get a steroid injection, you get a cortico injection, they make you space out how many you can have, and they limit the number that you can have. It's also why they do not treat it as a cure. It's not a cure for injury. Sometimes what happens is you can get an injection that will limit the amount of inflammation. Corticosteroid is going to inhibit the inflammatory agents. So let's say that you tore something in your knee. It's really inflamed. It's really painful. You can get a cortisol injection. It will temporarily inhibit that inflammation, and that may be enough time for your body to start repairing itself. So that way, then once the cortisol injection wears off, your body has already kind of gotten a head start on repair. If not, though, you are going to come back right to the same pain. Your injury is going to come right back, and you're going to be begging your doctor for another injection, and they're going to push you to have surgery. That's another conversation. Okay, disorders in adrenal cortex hormone secretion. Cushing syndrome, you may have heard of some of these. Cushing syndrome is chronic exposure to excessive glucocorticoid hormones in people taking corticosteroids for therapy. Again, therapeutic doses of cortisol. Some cases when the adrenal gland produces too much of the hormone. Obesity, hypertension, excessive hair growth, kidney stones, and menstrual irregularities are some of the symptoms. Addison's disease is a form of adrenal insufficiency. So whereas this one is excessive, this one is insufficient. Glucocorticoids is sometimes mineralocorticoids. May develop from a lack of ACTH or response to ACTH, or lack of response to it. Weight loss, fatigue and weakness, hypotension, and skin darkening. So you can see very much kind of opposites of these. And therapy of oral corticosteroids can hopefully treat it. Adrenogenital syndrome, congenital adrenal hyperplasia. It begins in the embryo or fetus. The inability to synthesize corticosteroids leads to an overproduction of adrenocorticotropic hormone. High ACTH causes increased size of adrenal glands. So increased size of adrenal gland, hyperplasia. Plasia is growth, hyper is excessive. And production of hormone with testosterone-like effects. And so it masculinizes the newborn. And let's look at the stress response. This is an important one to look at, especially as we are in this nice stressful time of life, and you chose to take an 8-week class. Thanks. So stressors elicit a stress response. The hypothalamus initiates this neuroendocrine response, and there are 3 stages to it: the alarm, the stage of resistance, and the stage of exhaustion. The alarm reaction is your initial response involving sympathetic nervous system activation, epinephrine, and norepinephrine. You see a big dog running towards you, and you kind of freak out a little bit. That's that alarm reaction, that first jolt of energy that's going to allow you to respond to stress. The stage of resistance is after depletion of glycogen stores. The adrenal gland secretes cortisol to raise blood sugar and help meet energy demands. This can be minutes to hours later. Stage of exhaustion is after weeks or months. Depletion of fat stores results in protein breakdown for energy, leading to weakening of the body and illness. So the stage of resistance will kick in once your glycogen stores deplete. So let's say that dog decides to chase you, and then maybe it's miles, and your glycogen stores start to deplete. You'll start to enter the stage of resistance where your adrenal glands will start to secrete cortisol, and that will start to do all of those things that we talked about. Where was it? Yeah, here. Cortisol will start to do all of these things to help increase your blood sugar levels to meet energy demands. And then if that keeps going on, so let's say that it wasn't just a dog tracking you, it was, I always like to use this as a moment to kind of remind people, we live very stressful worlds, we live very stressful lives. This is absolutely one of those times. So learning how to manage your stress really is important. Learning how to organize your work, your life balance, having personal time for yourself, not just, doing a bubble bath and putting on a face mask, but really being able to process your stress, process the things you go through. It's been shown that people that have PTSD don't actually enter REM sleep as deeply. And without REM sleep, we can't clear traumatic memories that we develop throughout the day and throughout our life, which is why people with PTSD tend to have bad flashbacks and difficulty sleeping and typically have to receive some type of mental health. So not being able to process your stress inhibits your sleep, and that just in turn increases all of these problems that we've been seeing. So stage of exhaustion, once you get to weeks or months depleting fat stores, your proteins start to break down, start to get weakening the body, you become more susceptible to illness, and essentially it's your body breaking itself down. So stress management is important. Enough of that. Okay, getting into the pancreas. So the pancreas sits behind the stomach between the duodenum and the spleen, so it's about right here on this side. You have acinar cells that generate exocrine secretions for digestion. So exocrine secretions, not endocrine secretions. These are going to go directly into the duodenum, into the small intestine. They make up the vast majority of the pancreas as acini. So the acinar cells are not endocrine cells. These are exocrine cells. The pancreatic islets, the islets of Langerhans, contain clusters of endocrine cells. You have your alpha cells secreting glucagon and your beta cells secreting insulin. I always just remember because there's an A in glucagon and alpha starts with an A. So that's what I remember. So alpha cells secrete glucagon, beta cells secrete insulin. There are also these things called delta cells and F cells that are present that secrete somatostatin and pancreatic polypeptide. We won't get into those as much. Here is a nice visual representation of it. And here we are. So the pancreatic hormones help maintain blood glucose. Normal blood glucose range is 70 to 110 milligrams of glucose per deciliter. High levels damage the blood vessels and kidneys. High levels of glucose will. Low levels will cause lethargy, mental and physical impairment, and death. Lowering high blood glucose levels with insulin. So after food intake, your beta cells detect the rise in blood glucose and respond by secreting insulin. I will let you tell me what type of stimulation this is. Insulin travels through blood and randomly leaves. You don't actually have to tell me, by the way. Please don't send me emails. Just answer the question right here on your own. Insulin travels through blood and randomly leaves the bloodstream to encounter target cells. And then insulin binds to receptors and initiates the second messenger systems once blood glucose falls, beta cells stop secreting insulin. So you have this negative feedback happening. Your hepatocytes remove glucose from the blood, stored as glycogen. Glycogenesis is stimulated. glycogenolysis and neogenesis are inhibited. Your adipose cells decrease fatty acid levels in the blood to help store fat, so lipogenesis is stimulated. And most body cells increase nutrient uptake in response to insulin. They also increase their amino acid uptake and protein synthesis, increase glucose uptake by incorporating more glucose transport proteins into their plasma membranes. And with less alternative fuels available, more body cells use glucose. So as your adipose cells are storing more fat, the other cells have to go to glucose. Some cells do not require insulin to take in glucose, like your neurons, your kidney cells, hepatocytes, and your red blood cells. There is a nice little regulation of insulin from the stimulus to its effectors. So clinical conditions, you've probably definitely heard of these. Diabetes mellitus is an inadequate uptake of glucose from the blood. You have chronically elevated glucose, blood vessels are damaged. The leading cause of retinal blindness, kidney failure, and non-traumatic amputations in the United States. It is associated as well with heart disease and stroke. Your type 1 diabetes is an absent or diminished release of insulin by the pancreas, so your pancreas is not releasing enough insulin. It tends to occur in children and younger adults, may have autoimmune component, and requires daily injections of insulin. Your type 2 is decreased insulin release or insulin effectiveness. Its typical cause is obesity. It tends to occur in older individuals or in younger adults. Treat with diet, exercise, and medications. Your gestational diabetes is seen in pregnant women, some pregnant women, not all. If untreated, it causes risk to fetus and increases delivery complications. It increases chance of later developing type 2 diabetes. But it can, and I believe typically does, go away after pregnancy. And then hypoglycemia. So your these are all hyperglycemic conditions. Hypoglycemia, blood levels below 60 milligrams per deciliter. Lots of causes: insulin overdose, prolonged exercise, alcohol use, liver kidney dysfunctions, deficiency of glucocorticoids or growth hormone. Symptoms: hunger, dizziness, confusion, sweating, sleepiness. It sounds like anyway. Glucagon given if the individual is unconscious and unable to eat. Glucagon will again help to raise blood glucose levels. So how do we do that? So when blood's glucose levels start to drop, the alpha cells detect that and release glucagon. Glucagon also acts through membrane receptors and secondary messengers, causing the body cells to release stored nutrients into your blood. So remember, it's going to do just like the insulin did, activate the G protein and some kind of enzymatic cascade. The hepatocytes release glucose. You have all of those activities being taken place, all the genesis and lysis that needs to be happening. Adipose cells releasing fatty acids. Glucagon does not affect protein composition, so it's not going to break down proteins or anything. And in glucagon can be given by paramedics to unconscious individuals with low blood sugar. And once blood glucose rises, glucagon release is inhibited, so it is also negatively feedback. There is that. All right, your pineal gland. Almost there, almost finished. The pineal gland is a small unpaired body in the epithalamus of the diencephalon. So again, if you are totally up or remember your brain anatomy, you can just refer to that. So your epithalamus above your hypothalamus. It secretes melatonin at night, causes drowsiness, regulates the circadian rhythm, has an effect on mood, generally a good one. And it influences the GnRH secretion, the gonadotropin releasing hormone secretion. Your parathyroid glands are small structures on the back of your thyroid gland. So again, the thyroid gland is on the front of the throat. The parathyroid glands are on the back of it. There are between 2 and 6 of them, usually 4. So they're just these little dots, these little pink dots, and they're just along the back of either lobe of your thyroid gland. They contain chief cells and oxyphil cells. Your chief cells, or your principal cells, make PTH, your parathyroid hormone, that increases blood calcium. It liberates it from bone, decreases it in urine loss, and activates calcitriol. If you remember the parafollicular cells in your thyroid, so these are parathyroid cells, they make parathyroid hormone that increases blood calcium. Your parafollicular cells in your thyroid gland are going to make calcitonin, which decreases blood calcium. So they have opposite effects here. Your thymus is epithelial cells that secrete thymic hormone. It's located anterior on top of the heart, so just on top of the heart here. It grows during childhood, but shrinks during adulthood and is mostly replaced with fatty tissue by adulthood. The maturation site for T lymphocytes within the blood cells, we'll get into those more when we do blood, chapter 18, and immunology, like chapter 22. Your endocrine tissue in your heart secretes atrial natriuretic peptide, which is ANP. So we're skipping now. So we did thymus has thymic hormones. Your heart secretes ANP. ANP is a hormone that lowers your blood pressure. It's by stimulating the kidneys to increase urine output and blood vessels to dilate. It essentially will detect the stretch on the heart. So as the heart fills with blood, it stretches. If it fills with too much blood, it'll stretch a little too much, and it'll start to release ANP, which tells the kidneys to increase urine output to decrease blood volume. Your kidney endocrine cells release erythropoietin, EPO, which occurs in response to low blood oxygen, and EPO causes increased red blood cell production. We'll get into that when we do, I think that's also chapter 18. I don't think it's right here. No, chapter 18. Your liver secretions include insulin-like growth factors. Remember, growth hormone stimulates that, and the inactive hormone angiotensinogen. Angiotensinogen is converted to active angiotensin II by enzymes from the kidney and the lung blood vessels by circulating the bloodstream. It helps to eventually raise blood pressure, causing vasoconstriction, decreasing urine output, and stimulating thirst. So liver secretes angiotensinogen. Eventually, it helps to increase blood pressure. Your stomach secretes gastrin that increases secretion and motility in stomach for digestion. As you might expect, gastrin is going to increase that mobility. Here is the renin-angiotensin-aldosterone system. Don't worry about getting too into this right now. Essentially, I just want you to know that you have angiotensinogen released from your liver. That is converted to angiotensin 1. Then it gets converted to angiotensin 2. And then it has all of these effects to help raise blood pressure. Like I said, once we do the next couple of chapters, we'll get into this more. But I just want you to go ahead and see it because it's something that will come back up over and over again. All right, structures with an endocrine function. Your small intestine secretes secretin and cholecystokinin into the blood, so two different hormones, secretin and CCK. Secretin stimulates the secretion of bile and pancreatic juice. Bile is going to help to alkalinize the acidic chyme coming from the stomach, and pancreatic juice is going to be a mixture of enzymes to help digest macromolecules. And the CCK, the cholecystokinin, stimulates the release of bile from the gallbladder. So secretin is going to be stimulated by the low pH from the acidic chyme. CCK is going to be stimulated by the fatty content coming from the stomach. And your skin cells, light converts modified cholesterol to vitamin D3, which is then released into the blood, so creating vitamin D. Calcitriol is the active hormone that raises blood calcium eventually. Your adipose connective tissue secretes leptin. Leptin controls your appetite by binding to neurons in the hypothalamus. Lower body fat is associated with less leptin, and this stimulates your appetite. So as body fat drops, you make more. It has other endocrine effects. Excess adipose raises the risk of cancer, delays male puberty. Low adipose interferes with female menstrual cycles. This is why, especially like for gymnasts, that can help to delay their menstrual cycles. Endocrine changes with aging. Secretory activity wanes with aging. It reduces efficiency of endocrine system functions. You have decreased levels of normal hormones, such as your growth hormone or your sex hormones. Reduced levels of growth hormone lead to weight loss and body mass. All right, and that was it.